Hello everyone, and welcome to this, our final webinar for this year. It's great to see so many people join us at a very busy time of the year. So first I'd like to start by respectfully acknowledging the traditional custodians of the lands on which we meet and pay our respect to elders past and present. Today, I'm going to give a very quick introduction to childhood dementia for those new to this area. And then we will have 3 speakers, as you will have seen when you registered, and I will introduce them properly just before they speak. Then we will have the panel discussion and question and answer. That will include all speakers plus Joe Kershaw and Professor Jennifer Byrne. You can write your questions in the Q&A box at the bottom of your screen. It looks like this in the corner. So for people who don't know me, I'm Chris Elvidge, Head of Research at the Childhood Dementia Initiative. And what I'm going to talk about, we've spoken about in length at previous webinars. So if you'd like more information, please feel free to go to our website and listen to our previous webinars. So childhood dementia affects 1 in every 2,900 babies, and it is caused by over 100 genetic conditions. And 50% of children with dementia die by the age of 10, and 70% will die before adulthood. Now, this devastating death toll is caused by not having enough approved treatments or enough clinical trials. So more research is needed. In August this year, we did a survey of childhood dementia researchers. And as expected, the #1 priority to accelerate research is, as expected, more funding. But also, the vast majority of researchers also said that access to patient samples and data such as natural history data would enhance their ability to do more childhood dementia research. So this topic is very, very pertinent, and I'm really looking forward to hearing from our speakers. Now I'm going to attempt to run a poll, and this poll is really for the researchers and clinicians in the audience, and your responses are anonymous, so please feel free to take the poll, which will help us with next steps. And I know that there's a diverse audience who have registered for the webinar, including families directly impacted by childhood dementia, government and industry representatives. So since this poll's not really relevant to you, please feel free to write in the chat, introduce yourself, say why you're here and what you're hoping to learn. And you can continue to put that in the chat as the webinar goes on, and I can review them afterwards. That will also help in our next steps. That's great, thank you. For everyone who's done the poll, I can see that blood samples are very much needed. 65% of the researchers have said that they would like to be able to access blood samples, also skin and brain tissue. About 50% have patient samples that can be shared with other researchers. 44% have patient samples that they can't share for various reasons. 28% don't have access. And some people have put some comments in so they can be reviewed later. Okay, I am going to end the poll now. results. Okay, stop sharing that. And stop sharing my slides, because now I have great pleasure in introducing Associate Professor Markus Hofer, who has more than 20 years of experience as a clinical neuropathologist and researcher. He is the consultant neuropathologist for the New South Wales Tissue Resource Centre and Deputy Chair of the Brain and Body Node at the Charles Perkins Centre at the University of Sydney. And patient samples have been pivotal to Markus's research to understand a type of childhood dementia called Aicardi-Goutières syndrome, research which has recently led to the development of a potential therapy. So please take it away, Markus.
Thank you very much for the introduction for that one. Can you see my slides?
Yes, great.
Thanks. Perfect. Yeah, thank you very much for the kind introduction, Chris. As you just mentioned, I'm primarily a basic researcher interested in trying to understand the causes of childhood dementias, but also trying to identify new potential treatment targets and treatment options. And the disease I'm primarily interested in is Aicardi-Goutières syndrome, and I'm going to talk a little bit more about it in a minute or so. But before I start with that, really first would like to emphasize of what I think is the most important part in research, and that is collaboration. And with that, I mean not just collaboration between researchers and scientists or researchers and clinicians, but actually the collaboration between researchers, clinicians, patient carers, but also advocacy association, advocacy support groups. And I think this photograph here, which was taken about 2 years ago at the Global Leukodystrophy Initiatives meeting in Philadelphia, really summarizes this collaboration in a very nice way. And we got here, for example, Devon and Patrick, two carers of patients with Aicardi-Goutières syndrome. So this is actually Rafaela, Devon's daughter, who was diagnosed with AGS. Both of them are also executive directors of the AGS Advocacy Association. And we met there at the meeting together with clinicians and researchers and these patient advocacy groups, really to try to identify what are the most important questions in the field and how we can target research really to try to address those questions effectively. So collaboration really, I think, is at the heart of all forms of research, but in particular, when it comes to rare diseases, where there's very limited access to patient samples and very limited number of researchers and clinicians focusing on those diseases. So back to AGS, or Aicardi-Goutières syndrome, as Chris already mentions, it's one of the causes of childhood dementias. It is a genetic disorder, and it can be caused by mutations in one or more of a group of at least 9 different genes. And despite this genetic heterogeneity, and the causes of AGS, what is common to all patients is the chronic overproduction of a protein called interferon alpha in the brain. So interferon alpha is typically produced in response to virus infections, and it's actually an essential protein in stimulating inflammation and immune responses against viruses. However, in the case of patients with AGS, interferon alpha is excessively produced, and it's produced chronically in the brain, where it then triggers a chronic inflammation, which ultimately leads to neurodegeneration and tissue destruction. Now, similar to the genetic heterogeneity, AGS is clinically quite heterogeneous. Patients can experience a wide range of different symptoms. Onset of disease is typically in young childhood, and key symptoms include profound developmental delays, but also developmental regressions. Patients can have a variety of different physical impairments and also a range of different neurological changes. So we in our research, as I said, very interested in trying to really understand the causes for AGS. And we've just recently published a study where we were able to identify the brain's blood vessels as a key mediator of disease in AGS. And for this study, we used a combination of mouse models, but also of tissue samples from patients and control subjects. And really the key findings are summarized here in the schematic. And basically what we were able to show is that the chronic response to interferon alpha in the brain induces a microangiopathy, that is a disease of the small blood vessels in the brain. And this disease is characterized by an activation of the endothelial cells that form the blood vessels. And it's also characterized by a breakdown of the blood-brain barrier, which normally separates the brain from the peripheral tissues. Now, this breakdown of the blood-brain barrier, this leakiness of the blood-brain barrier, allows the migration of toxic molecules into the brain tissue, and it also facilitates the infiltration of immune cells into the brain tissue. So other features of this microangiopathy also include the vascular malformations, such as microaneurysms, but also characteristic calcifications. And we are further able to demonstrate that actually this microangiopathy precedes the development of a more broader neurological disease and a more broader neuroinflammatory disease, which then ultimately leads to neurodegeneration. And we believe that this finding is really important because it, for the very first time, identifies a structure in the brain, that in this case, the blood vessels, that could be used as a therapeutic target for the disease. In the case of the blood vessels, that's obviously very important because blood vessels are much easier treated or accessible, much easier accessible to drugs than the brain tissue itself. Now, the second study, which I very briefly want to highlight, is a collaboration we are having with Ionis Pharmaceuticals, a pharmaceutical company in the US that specializes on the development of antisense oligonucleotides as treatments for diseases. And in this collaboration, we were using antisense oligonucleotides which inhibit the signaling of interferon alpha, and we use these antisense oligonucleotides in our mouse model. And very briefly, what we were able to show in the study is that these antisense oligonucleotides are able to not only inhibit interferon alpha signaling, but that these oligosense nucleotides actually also are able to improve neuropathology and the overall well-being of the mice. And therefore, we believe that these antisense oligonucleotides are really a very promising novel drug candidate for the treatment of this disease. And currently, Ionis Pharmaceuticals is considering the way forward, and they're planning for a phase 1 clinical trial to hopefully commence next year. So, but I think the point I want to make here with these two studies is that the use of patient samples is absolutely critical for us, for our research, really to identify potential drug targets, but also to test potential drug targets in samples. But on the other hand, it is very difficult for us to identify and to obtain access to patient samples. And the reasons for that is obviously that AGS, like many other diseases in the spectrum of childhood dementias, are very rare diseases. But obviously the genetic heterogeneity of AGS also further contributes to the difficulty of finding the samples. In the case for the study, the first study that I showed you about the blood vessels, we were able to include tissue samples from 7 patients. And it actually took us about 2 years' time to obtain those tissue samples, because only 2 of those samples actually coming from a brain bank, and the other 5 samples actually come from private collections and collaborators, and it was really tricky to get those tissue samples. On the other hand, I believe that tissue samples are absolutely important, in particular at this current stage of our research, because we are now at a point where several new drug platforms have been developed over the years, which are able to target interferon signal and may potentially be promising candidates for treatment, not only antisense oligonucleotides, but also another few small molecules inhibitor. And really, before we can translate the findings from the mouse model into human clinical trials, it would be really absolutely important for us to be able to really use tissue samples as a proof of a principle in that concept. So having the ability to access patient samples would mean for us that we could fast track those translational research efforts. So why is it hard to get access to samples? In addition, obviously, to AGS being a very rare disorder, the main reason really being that there is no existing network which connects different brain banks across the world. There's not a single database which would allow us as researchers to access and search existing brain banks for particular samples. There's also no consistency in the process, in the processes that are used to bank samples, also the type of information that is associated with particular samples. And very importantly, also that in most cases, patient advocacy groups are left out of the conversation. And I think that is a really big mistake, because patient advocacy groups provide the essential link between researchers and clinicians and the patients, and therefore to the tissue samples. Patient advocacy groups really would be in the position to communicate, to form the communication link between us researchers and patients, why it would be important, why it would be critical for us to be able to obtain samples, either blood samples, but also tissue samples, to really bring forward critical research. Now, if you look, for example, now at Australia, Australia currently has 4 brain banks, each one of which operates in a largely independent and fragmented way from each other. There's, again, no combining database which would allow us to search for samples. The funding to these brain banks is mostly ad hoc, and the brain banks are typically focused on very particular research areas, typically common adult diseases such as Alzheimer's disease or multiple sclerosis, and really do not include the collection of rare tissue samples. Again, another big deficit in existing brain banking. And finally, brain banking, particularly in Australia, relies on volunteer work, especially for neuropathological expertise. So in summary, I strongly believe that the system of brain banking, which is currently globally existent, is neither sustainable, and it's definitely not effective, and particularly not for childhood disease. And it's really beyond now the stage where we need really to consider new ways of bringing forward brain banking, tissue banking, to make efficient use of these really most valuable tissue samples that are donated by patients in our research. And I think that's it from my side. So thanks very much for the invitation, Chris.
Thank you, Markus. That's great. So now I have great pleasure in introducing Associate Professor Greg Sutherland. He's a research group leader in neuropathology, Director of the New South Wales Brain Tissue Resource Centre, the Brain Bank, and Associate Professor in Neuroscience in the School of Medical Sciences, based in the Charles Perkins Centre, University of Sydney. And Greg is spearheading, along with Markus, an international virtual rare disease brain bank initiative to enable the sharing of brain tissue for paediatric brain disorder research. And I look forward to hearing more about it. Thank you, Greg.
Thank you. Thank you, Chris, and thank you very much to the Childhood Dementia Initiative for the kind invite to speak at the webinar today. I'll just share my slides and check that everyone can see those. Can you see my slides?
That's good. Thanks, Greg.
Okay. So, yeah, look, as pointed out by Chris, Markus and myself have developed the idea basically from his experience with AGS and some of my own experiences of attempting to establish a resource for helping researchers who are interested in the childhood dementia diseases. And I just want to talk, following up on what Markus said about a little bit about this initiative, and from my point of view, how I got to the sort of the same place where Markus is. So just a quick disclosure, as a director of the New South Wales Brain Tissue Research Centre, I just wanted to note that we're funded by the National Institute of Alcohol Abuse and Alcoholism, one of the NIH institutes. So just a little bit of an outline, what I'm going to talk about today. I'm going to talk a little bit about background very briefly. I then want to talk about biobanking, and particularly brain banking pipelines. I'm then going to speak briefly about some case studies, including one from the Childhood Dementia Initiative that was instrumental in the way we're thinking about the solution that I want to present to you, and then the solution itself, an online registry to internationalise brain banking for the purposes of rare disease research. Okay, so the why, and most importantly, we all meet, particularly the people on the webinar today, will be all familiar with the patients and families and the desperation to find cures, and also the frustration that things don't happen quicker for these precious individuals. And then secondly, as Markus was going into, researchers really struggle to get enough tissue to do the type of science that you, as a researcher, can be satisfied that what you're finding is likely to be real and not occurred purely by chance, i.e. finding sufficiently large cohorts to work on, but not only sufficiently large, ones that have sufficient clinical data because of what Markus was saying with the heterogeneity that's often inherent to these diseases. So there are 4 brain banks in Australia, and as Markus was saying, they deal often from a point of view of demand on common diseases. These include the dementias and Alzheimer's disease. And a typical pipeline would be that the brain banks associate themselves with tertiary referral clinics. So if I use Parkinson's disease as an example here, there may be a movement disorders specialist who will receive at their referral clinic patients who have initially seen neurologists. And so it acts as a way of collecting together a large number of individuals in a sort of a one spot. And it's often then that it is that particular clinician who will have the early conversation with people and say, would you consider brain donation after your death for the purposes of trying to improve our understanding of the condition? And the advantage for the clinicians of having that conversation and getting their patients to donate their brain after their death is that in the first instance, neuropathologists like Markus are able to go through the brain and confirm that the disease that they clinically thought the individual had was indeed the case. Many of these diseases are only diagnosable at autopsy, sorry. But the other advantage is once that tissue is collected, then that tissue can also be sent out to researchers around the world who can apply a number of different techniques where they have expertise and collectively try and understand these diseases a lot more. And that's essentially what my brain bank and other brain banks in Australia do. So when we get a donated brain, there's an autopsy done to retrieve the brain. And then from there, it's sort of a brain of 2 halves. So standardized processes to cut the brain down the center, and half of it we fix and the other half is frozen. And the reason for this is the techniques that are applied by researchers can generally be divided into what is done on fixed tissue and what is done on frozen tissue. And as I'll show you shortly, we're now starting to combine those two techniques and also to combine them directly. But typically at the moment, my brain bank is based at the Charles Perkins Centre, which is on the Camperdown campus of the University of Sydney. And our facilities are shown here, and we bank tissue. We have just a little bit over 500 individual brains, and that is banked in terms of half of those brains are cut up and banked in freezers, and the other half is held in fixative. And then right on the right-hand side on this particular slide is a typical section of human brain tissue that has been sliced extremely thinly down to around about 7 microns or 7/1000ths of a millimeter, and that tissue is stained. We refer to that tissue as formalin fixed paraffin embedded tissue, and to that we apply various antibodies or potential targets that we think might be important in the disease. And this is the type of stuff that we can get once we've stained up the tissue. The left of this image is a picture of a part of the brain called the cerebellum. And this is taken from a person with Alzheimer's disease. And those orange sort of pock marks that you can see on the background of red there, they're actually amyloid plaques. From the other side of the brain that we freeze, one of the modern and current techniques is something called single-cell RNA sequencing. And this has been a real revolution in brain diseases, because rather than considering the brain in terms of microglia, neurons, and astrocytes, we now realize that of all of those type of different cells, there are subtypes. And those subtypes include microglia that express the interferons that Markus was talking about as an example of one microglial subtype. Now, these two technologies have recently come together, and I'm just showing you some variations on a new technique called spatial transcriptomics. And the one up the top was the original type of spatial transcriptomics. And what you're looking at there with all of those spots is the expression of cells within the brain. The green right up the top there is indicative of a white matter. And then below that, as a case may be, is the different layers of a human grey matter, where for most of our brain, we have about 6 different layers of grey matter. But now with the increase in the resolution of these techniques, we can actually go down to single cell level, and all the sort of yellow spots, what you're seeing here, are a protein called myelin basic protein. And the reason why the signal is very strong in the middle is that this is white matter separating 2 parts of grey matter in a human brain sample. And white matter has concentrated with cells called oligodendrocytes. And the oligodendrocytes make the myelin, which is the fatty substance which insulates the nerves as they move from the brain off to other areas in the brain or to the spinal cord. And so myelin basic protein, which sits in the myelin, is highly expressed in white matter. And I'm just using this as a case to show you the sort of technologies that now can be applied to brain tissue. And these have only really come up in the last couple of years. But what it has meant is that there's a lot of excitement, again, about using human brain tissue, because we've just got these amazing techniques where we can visualize what's happening right down to a cell level. Okay, so sorry about that. So I talked about the brain banking pipeline, and Markus talked a lot about this idea that not only is the tissue important, but of the clinical information. You may also have mentioned that it's not always the case where information or research that is done by researchers where that information actually gets back to the donors and the families, or even occasionally the clinician. But typically we envisage pipelines as sort of linear structures. But when I started thinking and preparing the talks that I've sort of done this year on brain banking, I started to think more that the pipeline is really circular, because the tissue really starts with the donors and their families, and yet the information is that we want to get back is it back to the families via their clinicians. So it's very much a circular pipeline. I talked about that brain banks at the moment are very much work on common diseases. There's a number of reasons for that, and one of the major ones is funding, but also that we can more easily get together sufficient cases in one brain bank to be able to have a researcher come to us, and we act as a one-stop shop to supply them with sufficient tissue to do their research. But what happens when you have rare diseases? Well, it takes a little bit of thinking outside the square. You have to kind of consider, well, where are we going to get our donors from, and how often are those donors going to come up, in an area within Australia, including some of the metropolitan cities. So to try to illustrate going forward in this area and thinking outside the square, I wanted to use two quick case studies. The first one had been the subject of a recent Australian Stories, and it involved the Snelger family and their daughter Blair. And unfortunately, a couple of years ago, Blair passed away. She was only 3 years old, and she suffered from Angelman syndrome, which was actually even very young for someone with this particular syndrome. Very sad story, but it was the wish of Blair's family that her tissue could be donated to research. But Australia wasn't set up for banking rare diseases. However, through the Cure Angelman Foundation and a lady, Megan Cross, who's based up in Cairns, she was aware as the member of the advocacy group of researchers who were working with human brain tissue from Angelman syndrome, and indeed were wanting to start a brain bank in near Sacramento at the University of California in Davis. And so she came to me and said, young Blair has passed away. Is there some way that we can get the tissue to America? And my first thoughts, given this regulatory environment and also ethics in this area, was that it probably wasn't going to be possible. Nevertheless, I did some inquiries, and it turned out that at least at the University of Sydney, that they were happy that the New South Wales Brain Tissue Resource, or our brain bank, could act as a conduit. And so we were able to send not one Angelman syndrome donated tissue over to America, but actually two in the last few years. And also that was through the help of another brain bank run by Associate Professor Michael Buckland. So based on those experiences, I started to think, well, hang on, here's a real opportunity for us as brain bankers to maybe add value. We might not keep these collections ourselves, but we can certainly move them to areas where the best research can be done, and potentially centralized collections. So the second quick case study I wanted to talk about was a very own childhood dementia initiative, and this idea of under one roof, being able to capture all of these diseases. In the case of the CDI, this allows them to lobby and fundraise on behalf of all these different rare diseases. And we really wanted to take sort of advantage of this type of idea and say, okay, well, how can we use this similar approach to brain banking in rare diseases? So I'll quickly share this particular slide, but it really what it talks about is the different stakeholders, including the brain banks and researchers, and the advocacy groups, and really outlines what everyone is trying to achieve. And there's a lot in common between all of us, but we all have slightly different needs. And so taking in all of these requirements, we started to realize that, and basing it on the plan of this circular sort of pipeline, that there were a number of synergies between these different groups that was going to be possible to capture in a, or could be captured if we set up the right type of plan for brain banking, which I'm going to talk about in the next slide. And these are the type of synergies that I referred to with the case studies. The fact that advocacy groups may know or may have even fundraised for particular researchers who would be unknown to the brain banks, but also that advocacy groups might consider that brain tissue donation is also a valuable way of stimulating research into their disease of interest. Okay, so this is where Markus and I and a few of our colleagues came to. We decided that the best way to go about this was a virtual brain bank. A virtual brain bank being a registry, trying to keep our costs down. Initially, we thought it needed to be web-based, voluntary participation, and then the idea was that by initially creating a registry where we knew from around the world, given that we're talking extremely rare disease on many occasions, and in private collections, could we bring virtually all of those collections into one web-based database. And then we had ideas of how we could then go on from there. But I just really want to talk about that one idea today. We reached out to Mick Badron. Mick is an IT consultant, but he's done a lot of work in the non-for-profit space, and particularly in the health space. And we recognised that if we were going to start, even if we started small, we needed to start well. So any IT solutions we had, we wanted to be able to make sure that it could be added on to and scaled up. And so Mick has sort of come on board with Markus and myself, and between us and a committee, we're sort of moving ahead with the following, which we've called, or Markus came up with this a very interesting idea of calling our online database Brain Bank Connect. And we are at the moment running workshops and indeed connecting all the brain banks around the world. At the moment, we're up to about 60 individuals representing nearly 20 different brain banks, including the large brain banks in America and Europe. And we are just putting together our business plan, but also our database, which will look a little bit like this, or this is a very simple schematic, to be able to capture where around the world all these different cases for all of these rare diseases are. And we're hoping what this will allow us to do going forward is to act as a sort of, I suppose, not only a way of alerting researchers to what is available, but also to act as a sort of a lightning rod to stimulate research. So lastly, I'd just like to acknowledge both my own funding sources, but also the Childhood Dementia Initiative for putting on the webinar, the Snelga family, who have been really kind and generous through their Australian story, and also to give permission for me to use their sad experience to shine a spotlight on rare diseases. And lastly, Megan Cross and the Foundation for Angelman Syndrome, who have been very important in giving me insights into the plight of the advocacy groups and the families in this area. And I'll leave it there.
Thanks, Greg. That's great, and I'm sure we'll have some questions on that in the panel session. And of course, brain donation brings up a lot of ethical and emotional issues as well. that need to be considered, especially with donating post-mortem children's brains, and
Yes.
that's where the patient advocacy groups can come in.
Yeah, absolutely.
Yeah, definitely. Okay, so next I'd like to introduce Dr. Laszlo Irini. He's a senior project officer at New South Wales Health Pathology. He works on various biobanking projects funded by the New South Wales Office for Health and Medical Research, including a scoping project for childhood dementia. So go ahead. Thank you.
Hi. Oh, hi. Hi, Chris. Can you hear me?
Yes, that's good. Thank you.
Thank you. So, hi, thanks for the introduction and thanks for inviting us for this webinar. I would like to just give a short introduction on what are we doing here, what is our project, which is actually funded by the Office for Health and Medical Research of New South Wales Government, and started early this year. So the project what we are doing is a pilot and feasibility study to establish long-term follow-up system or clinical registry and biorepository for patients undergoing cell and gene therapy in New South Wales, which of course it includes childhood dementia patient too, because as we know, actually most of the people suffering from childhood dementia, the cause of childhood dementia is genetic conditions, and there are plenty of therapies in the pipeline of pharmaceutical companies and also clinical trials that cell and gene therapies, which actually gives hope for patients having rare diseases such as childhood dementia. So those therapies actually are coming to the market as we probably around like eight therapies are already available commercially and approved by TGA, in US actually even more, and plenty of clinical trials, but in the pipeline. But nobody knows actually what about the safety, efficacy, and then durability of this treatment. As the studies show so far, those approved therapies has very limited success so far, unfortunately. So it's very important that create a system, kind of system to long-term, which enable us that for long-term follow-up of those patients, for clear surveillance, and also to have a system like this to facilitate research, because it's very important that to have all the registry, the data, and of course, as we heard about this afternoon, that the samples, because that's the samples actually which give access for further studies, research, or if anything goes wrong down the pipeline 5-10 years with the therapy, there is some samples actually which you can go back and see what happened actually. So our project is to establish, investigate the feasibility of such a kind of virtual and biological register model for patients receiving CGT products, and also to create a kind of virtual platform network for sharing childhood dementia data and patient samples to accelerate research progress. And what we are doing all this to support local development, research, and post-therapeutic surveillance. It's very important to consider how those samples actually are banked, how the data linkage is actually performed between the banked samples and the registry, and what is the sustainability of such a kind of virtual and biological registry. As we all know, actually everything costs money, and the funding actually, unfortunately, is not available yet. So why is it important actually to create such a kind of system? Of course, as to investigate. Oh, sorry. To investigate what is the efficacy, safety, and durability of the therapies. So because Of course, there are many things that what we know, there are things that we know that we don't know, and what we forget we know, and what we don't know what we don't know. So for that, we need actually databases, and we need databases and data to answer those questions. And this is actually will support both therapeutic surveillance and new medical research. So as probably everyone is aware of the rare disease clinical registries in the field of childhood dementia and other rare diseases. So there are plenty of available, which mainly run by patient advocacy groups, clinicians, national health system, even pharma companies. But those actually all kind of individual registries and not really connected. So these registries actually can serve many purposes. What we are interested in actually in case of childhood dementia, of course, to support research. And for that, to support research, as we heard here, we need representative population and comprehensive databases. So we just heard about the example of the brain biobank network. So actually the technology, I think, is already available to connect data, whatever data we have actually, either in the registry, either in the biobanks. And there are plenty of software for that, network for that. But the really big question actually that how are we going to achieve this? So New Success has actually coming out a new patient record system, the single digit patient record system, which gonna facilitate largely to connect data in clinical databases and in research environment. Anything which is connected to the patient can be found using this kind of data. That's the kind of aim. And if I understand well, that's going to be implemented in the next few years in New Success Health. But again, even if the data is there, even if the sample is there, the biggest question and challenges comes that how to share those data, and actually those data and samples actually available for someone who has a great idea, research idea, and would like to access those samples to answer his questions. The biggest challenge is actually informed consent. Most of the patient samples in the biobanks are probably from patients actually who passed away, and the big question actually how the consent was written at the time. So it was like specified or unspecified research, or actually it needs to be reconsented with the parents. What about the ethics? Ethics usually it's very limited for certain research and not like unlimited research. What about the funding? Of course, it's always a big question how they're gonna fund such a kind of network. And of course, ownership, privacy, security, and standard of the data. As we know, different databases have different standards, such as the biobanks. So there's plenty of work to be done to establish such a kind of network of samples and data. So the biggest, what we would like to achieve is about the FAIR principle for sharing data and sample. The FAIR principle means findable, accessible, interoperable, and reusable. So we would like to create a kind of such a network for childhood dementia initiative to connect. It's possible to connect those samples and data available and for research purposes. But how to do that? That's actually the biggest challenge, and who's gonna find actually financial resources for that? I would like to actually just bring up some example of what happened actually in other fields. Maybe people are aware of this Aicardi-Connect, this Rare Disease Connect. They create a kind of platform when both the patient samples the registries connected with researchers to facilitate research. And this is just a kind of brief graphical summary of their work. That's something like actually what we would be possible to achieve, or what Jennifer Byrne did previously in his previous job. Her previous job is a biobank network. That's a great example. It's the Australian and New Zealand Children's Hematology Oncology Group Biobank Network. So that's it's still existing. It's a collaboration between existing biobanks. They got seed funding to establish this network from philanthropic organizations. They have a steering committee, and of course, their aim is to improve operational sustainability, sharing information, visibility, and recognition, improve sample access for researchers to establish a centralized access point, and researchers can request information about available samples through a biospecimen locator. So what are the next steps? What the next step actually would like to, first of all, we need to know what is available out there. So what kind of samples have and who have this? So I think a survey for childhood dementia research community need to answer those questions. What type of data samples do they have? Can these samples and data be shared? It's very important, the ethics, what is in the ethics, the informed consent, and who is the ownership of those samples and data, and where they are, I mean, physically, where are they located, how they stored, and what is the availability of those samples. So that's a very, very short presentation, what we are trying to do. I mentioned it's actually a feasibility study. We just got funding to investigate all those enablers and barriers. So, and to actually to proceed with such a kind of network, it needs both financial support and both support from clinicians, patients, researchers who actually agree to participate in such a network and work on this. Thank you very much for your attention. Yeah.
Thank you, Laszlo. That's great. Thank you to all three speakers. If all the panelists would like to turn on their cameras, that would be great. I'd also like to introduce Jo Kershaw, who is the Lived Experience Manager at Childhood Dementia Initiative. Her role involves listening to families directly impacted by childhood dementia, documenting their experience and amplifying their voices to make changes for all families. And Jo works with over 70 parents and bereaved parents of children with dementia who've joined our Family Advocates program. I'd also like to introduce Professor Jennifer Byrne, who's the Director of Biobanking at New South Wales Health and Professor of Molecular Oncology at the University of Sydney. And she's based at the New South Wales Health statewide biobank. Welcome to both of you. And thanks to all the panelists. I'd like to start off with a question for Jo, because we haven't heard from the family side yet today. Jo, what do you hear from families about participating in research?
Thanks, Chris. All the families who we work alongside and we hear from them, research is a priority, and research to develop curative and therapeutic treatments is a priority for them. So if all families not only want to be involved in some kind of research, they really express to us that it is their priority because they know that nothing will change without research and their families essentially, and this cohort of families participating.
Thank you, Jo. We are running quite behind on time, so I can see that Julia Warning has her hand up. Julia, did you have a question you wanted to ask live? I think you'll need to unmute. that's not working. Anyway, if you did have a question, Julia, if you want to type it in the chat, then I can ask for you. I'll go to some of the questions that are in the Q&A box. So a good one I saw come up from Falak was for Greg. Whilst we wait for all this incredible work to come together, how can we respond to families now? Do you have any advice for how families can drive banking of brain tissue should they wish to?
Yeah, thank you for the question. Well, I think, I suppose the thing off the top of my head would be that we really, and the reason why we've developed the brain bank connection was that we need to try to get sufficient amount of tissue in the hands of people who can actually do the latest science. It does matter. Well, you do need to have a good background in these diseases, as Markus demonstrated, to know where the likely hypotheses lie, or the likely targets are for therapeutic improvements. So, you know, we're really just keen to make that happen. So, for example, if a researcher or via a brain banker that they contact can come onto our website and get an idea of how much tissue is around the world, and potentially also that tissue can be held in one spot, although there are, as Laszlo was referring to, a number of international regulations in different countries that do not permit tissue to be transferred. But at first instance, we would like to just allow people like Markus to know, okay, well, around the world there's this much tissue that potentially I can use and to set up my experiments.
So for families, if they did want to explore brain banking, would they be able to get in contact with you, or would they be better off contacting their advocacy organisation?
Yes, they're very welcome to contact myself. But yeah, that would be the other option too. I mean, we're very young with this. Very sort of just beginning this initiative, so we certainly haven't reached out to all different advocacy groups that exist in Australia. But of course, using the CDI as an example, we've federated a number of these types of groups that allows us quickly to let people know that this is a possibility. So yes, they're most welcome to contact me, and we would then either look to move tissue where it can be done the most good, or we may even hold it ourselves in lieu of trying to establish those collections.
Right. And Jennifer, you haven't had a chance to speak yet. Is there anything you'd like to add after all the presentations?
Oh, thanks, Chris. I think there's a question, I think, in the chat, which is, do we need to make a rare disease biobank? Would it be better to have larger biobanks serve multiple functions and aims? I think it's a good point, around consolidation can improve cost efficiency. But I think in the case of childhood dementias, we probably need to think about what we already have and what we can do, and sort of start with the most pragmatic solution first. In the case of the childhood cancer biobank network that I set up, we didn't attempt to consolidate samples just because of the expense and complexity of that. We really thought that it was most efficient to have the samples where they were, but to promote collaboration between biobanks, use of best practices, and very importantly, to have a one application process for all biobanks in the network. So researchers only have to make one application, and then they can access the resources of multiple biobanks through that. So I think that there's a lot that we can do in a virtual networking context, as I think Greg's also given that example today as well. It's not always just about physical consolidation of samples.
Yeah, great. And I guess going forward, as well as we start to talk about centres of expertise around the country for rare diseases and for childhood dementia, whether we can think about prospectively collecting more samples and how we do that in a coordinated way.
Yes, I think so. And I think one of the really key things is thinking about consent, where possible, trying to make patient samples available for future unspecified research through the consent process. That can really enable sample sharing and sample reuse for projects beyond what might have been imagined when they were first collected. And that's certainly been invaluable in the childhood cancer space.
Okay, great. Thank you. Unfortunately, we're really out of time. There's been a couple of other questions, great questions in the chat. One about banking eye tissue and other about rare disease coding. So they're both great comments that we'll take on board and share with the speakers and answer. I just would like to just share my thank you slide. And that's working, yes. I would like to thank our speakers and panelists today for your time and your presentations. And if you have any further questions, feel free to reach out to me. My email address is on the screen there. You can also head to our website for more information on how to get involved for families, researchers and health professionals. Thank you to Illumina Corporate Foundation for supporting this webinar series, and we will have the final one in this series early next year. And as always, thank you to the families who give such valuable input into all the work that we do at Childhood Dementia Initiative. Thank you all, and we will see you next time.
Thanks very much. Thank you.
Thank you.
Yeah.